Let's get started. Let's settle down. Okay. Thank you very much for coming. And I actually didn't expect this big turnout. And my name is Yunlong Liu. I'm the director for the Center for Computational Biology and Bioinformatics, and also Center for Medical Genomics that runs this 10x instruments. And for the past half a year, or a little bit less than half a year, since we officially acquired the 10x instrument, so far that we have done the single-cell RNA sequencing experiment for over 80 samples. And that is really an incredible run, and considering how expensive these assays are. You guys need to work on that to reduce the cost a little bit. And so during this couple of months, through the various data analysis, and surprise, we found out that the most important factor for successful run is the quality of the cells that you guys provide to us. If all the cells are dead, the chances we're not going to get very good data out. And we also know that this is not the strength of our center in terms of cell biology, because every group is working on their own stuff for decades, and there's no chance for us to understand how to deal with different type of cells and how to make them healthy and happy. But at the same time, I know that most of the labs are struggling on this in terms of how to best fit the requirement of the 10x, and also other type of sequencing instrument that more involve the cell biology. Because of that, and we are arranging this workshop, and we are very fortunate to have the 10x scientists and also Miltenyi scientists to come here and to teach us, to have this workshop on how to prepare the cells and making sure that your samples is most optimized for the single-cell RNA sequencing, and also other type of data analysis that we might need to run in the future. So we booked an hour and a half in this room for the workshop. We probably will take less than that. But after that, from 11:30, I believe, to 4 p.m., we have a R3 buildings 428 booked, and all the speakers will be here to answer the specific questions that your lab may have, because as I mentioned earlier, for different biological system, you will have a pretty unique problem that only your lab is facing. So, and we will just pick their brain and try our best to get as much as possible from them as possible. And another thing that I'm considering is if there is large enough demand, and we probably want these scientists to come here as for the office hours, maybe once every 2 months or 3 months, so that in the next couple of weeks or months, that when you play with yourselves, you met with specific problems, and then they will be able to help you to address that. So, but we will need to have enough demand for that. So the chances we probably will have a signing sheet on that, and so that to do better coordination. Okay, so without further ado, we will start from 10x. Thank you very much for coming. Thank you.
Good morning. Thanks again for coming. Thank you, Yunlong, for having us. So my name's Hadley. I'm the local salesperson for 10x Genomics. We're very excited to talk about this. As Yunlong set the stage, this is the most important part of the 10x assay is sample preparation. So a quick agenda on how we're going to run today is I'm going to talk very briefly about some of the new things that are coming in the coming months here. From 10x Genomics, it'll be very quickly, but I want to make sure everyone's aware of those things. And then we'll hear from Jim Casper with Miltenyi on his talk on sample preparation, from tissue to cells. And then we'll hear from Egon Rankini, our local 10x field application scientist, who will speak more specifically to single-cell sample preparation, our recommendations. As Yunlong said, we'll be here all day. If you want to connect with us a different time, please feel free to shoot me an email. We can connect and help answer any questions. Our job is to make you guys successful and make this as easy as possible. So very quickly, just to give those of you who don't know a little bit of quick background on why single-cell genomics. The main reason is because everyone knows how immensely complex biology is, right? We have 20 trillion cells, 6 billion base pairs, lots of things to interrogate. And at 10x Genomics, we are looking at that from the single-cell genomics perspective and continuing to create new applications that help elucidate these different complexities throughout our bodies. And so how we do that is through something we call GEMs, gel beads and emulsion. Hopefully you all are familiar with this by now, but this is the core of the 10x Genomics technology. We start with barcoded gel beads. We send them through a microfluidic system with your sample of interest, partition them with oil, and create these gel beads and emulsion. So that's the basics of the 10x technology. We have 4 product lines. We started in 2015 with something called linked read sequencing, looking at long reads for haplotype phasing. We expanded from there to single-cell gene expression, which is probably why you're all here today. We moved from a 3' gene expression product to a 5' immune profiling product to couple the single-cell gene expression with T-cell and B-cell repertoire. And then for the first product of this year, we released a copy number variation profiling assay at a single-cell level. So again, what we're trying to do at 10x is get a more complete picture at the molecular level, and you could think about the many different ways that we would try to do that in profiling cells based on different things, right? Genome, epigenome, transcriptome, et cetera. So the first new product that'll be coming that I want to make you aware of is based around the epigenetics portion of this goal, right? And so that starts with an attack-seq assay. I'm sure everyone's familiar with this, but this is an assay for transposase-accessible chromatin using sequencing. Essentially what we're looking at here is adding a transposase to nuclei to cut at certain sites and insert sequencing adapters into sites of open chromatin to profile those through next-gen sequencing. And so we took that idea and are releasing the first commercially viable solution for this assay. So you'll be able to look at up to 10,000 nuclei from a sample, define different cell types in kind of a new way, right? And so what 10x has kind of become famous for is pushing the boundaries on how many cells we can interrogate in a single sample. And so if you look at these pictures here are based on attack-seq profiles of more and more numbers of cells, up to 10,000 plus cells. I mean, as you can see, you quickly learn to resolve different clusters, different cell types with higher numbers of cell resolution. And so from there, you can use different transcription factor binding sites and motifs to classify these cells in different ways. And so that's what we're able to do with this assay, which should be released soon. And we're very excited to be able to bring this to you and see what you'll be able to do with it. So that's the first one. That'll be out very soon. If you're signed up with us at 10x, you'll be hearing about it in the coming months. And then the second thing that you've probably heard about by now, but we are also releasing, is looking at a few different of these little colored boxes here, but specifically with respect to the proteome and the transcriptome at the same time. We're building off a paper, or 2 papers that came out, CiteSeq and ReapSeq. These came out end of last year around the same time. CiteSeq from New York Genome Center and ReapSeq from Merck. Essentially what they're doing here is they're creating a synthetic mRNA that they attach to an antibody. and give that synthetic mRNA a barcode that can then be tied back to that specific antibody. So the idea here is you wash your cells over with these barcoded antibodies for your protein of interest, run them through 10x like you normally would, and then you're able to get both an mRNA library for sequencing and a protein library. And then interrogate both of those at the same time, at the same, with the same exact cells. Another example of using this type of technology is something called Perturb-seq, where you're using the same type of barcode idea to attach it to a guide strand of a CRISPR library to do massively parallel functional genomics, where you can transfect your cells with these barcoded guide RNAs and look at the different changes to the transcriptome that those perturbations have caused. And so what we're doing at 10x is enabling both of those types of technologies and many more. If you think about the technology, you can really just attach a barcode to many different things. I think a lot of our customers are pushing the boundaries here, and I think many of the people in this room will have great ideas around this as well. But we'll be enabling this from a commercialization standpoint, where you can easily do this, where you can add your antibodies and pull out both the gene expression and the protein targets and easily analyze it with the software that comes with the 10x system. So those are the two new things that are coming, single-cell attack-seq and what we're calling this feature barcoding thing. If you have questions about it, there'll be a lot of press releases and papers coming out soon, but please feel free to reach out to us if you have questions around these. So now, while you're all here for how to apply these cells to these different technologies, this is Jim Casper. He's your local Miltenyi representative, and he's going to talk about sample prep.
Thank you so much, Hadley. So, hi everybody, I'm Jim again. I am your Miltenyi biotech rep. So I'm going to go over sample prep and a little bit of cell isolation. It's really, really simple. I'm going to keep it really basic, really simple, so we can go through it quickly. And just to kind of educate you on our products if you're not familiar with them, if you guys are used to using home brews or anything like that. So basically, you have our sample prep GentleMax technology. You start out with solid tissue, and with our portfolio, you can start out with a number of different types of tissues: spleen, skin, fat, heart, liver. Doesn't matter. We have something for you. So, the goal is to start with, the solid tissue and then get to this downstream single-cell analysis like Hadley briefly spoke about. The research areas, depending on any research area you're in, as long as you are starting with human tissue or mouse tissue or rat tissue, you can, use our sample prep products. So a lot of researchers use homebrew methods that are challenging from user to user and can be inconsistent. A lot of researchers don't think about critical factors such as epitope preservation. Yes, you can get your cells and get your single cells out and get a nice homogenate, but you have no idea what's really happening to your cells if you're using your home brews. And you need to use reproducible and quality products for quality research. A lot of homebrew tissue dissociation is using mechanical ways to break apart the tissue and enzymatic digestion using some enzymes, which could be an issue when it comes to keeping your cells healthy. Some of the other mechanisms are just manual, mortar and pestle, homogenizers, and bead mills, bead beaters. I don't know how many of you have actually used bead beaters, but they really, I could put my arm in a bead beater and it would become homogenate in about a minute. So you really don't know what you're doing to your cells if you're using a bead beater. And then again, with Miltenyi's kits and our reagents, you have purified and validated enzymes. We know what's happening with the epitopes on the cells. We've done the research. And in fact, with some of our tumor dissociation kits, we have exactly what epitopes are left, like what epitopes are being destroyed or being left. But with crude enzymes using home brews, you're really destroying some of these epitopes that you might want to use further downstream. And I get a lot of questions or a lot of pushback. Well, I'm using my own home brew and my cells are just fine. That may be, and that's great. But if you ever want to pursue research further down the line, those epitopes might be gone, and you have no idea what to do then. So that's why I want you to come over to Miltenyi. So really quick, just to go over our sample prep portfolio, we have our GentleMax instruments. We have our C tubes and M tubes that are used within those instruments, and then we have the tissue dissociation kits. So it's really easy. So basically, well, I think I get into this here. Yeah. So so this is our GentleMax Octo Dissociator with heaters. So basically it has 8 different points that you can put the tubes in to use for tissue dissociation, so it can hold 8 samples, and they all move independently, so you can process 8 different types of samples. You don't have to have 8 samples of one, or 8 different types of one sample. You could have just you could have liver, you could have spleen, you could have brain, you could have anything on here. They all run independently. And then we have the heaters. The heaters go over the tubes and create a nice environment for the enzymes to work, because most of the time you need heat for enzymes. And then the C tubes are for our cellular, so cellular C tubes. So that's for saving those cells, making sure those cells aren't breaking apart. Our M tubes are for molecular. It's basically just breaking apart everything, so you can do your mitochondrial work, your protein work, your DNA and RNA work. And then we have we have our tissue dissociation kits and our smart strainers. Our smart strainers come in 30, 70, and 100 micron mesh that can kind of get rid of some of the cell clumps and some of the other stuff that you get when you homogenize tissue. And then our tissue dissociation kits, and then I'll have a list of all of our tissue dissociation kits. So the good thing about our tissue dissociation kits, in the protocols, depending on what type of kit you're using, we have the corresponding protocol in the instrument already. So if you buy our spleen dissociation kit, and it tells you to use our spleen program is already in the instrument when you purchase it. So it's a really nice, easy workflow for you to get through. And also we also have tissue storage solutions, so you can preserve some of your tissues if you want to dissociate a day later or something. We have solutions for the customers with that issue. So our mission statement pretty much for our sample prep technology, it basically enables simultaneous mechanical and enzymatic solid tissue dissociation in an optimized and automated format that is gentle, and that's the key word, gentle. The cells, you're not destroying their cells, you're not beating them up, you're not killing them, you're really gentle, and you're creating that nice homogeneity where you can move forward with your downstream assays. So this is pretty much what a tissue dissociation workflow looks like. You start your solid tissue, you just mince it a little bit, and then you throw it into our C tubes with our tissue dissociation kits. And it's a nice protocol, data sheets, and all the kits. You pick the program that you want, you throw it on there, and with our Octo, with heaters, it's all automatic. You just put your tissue in, put the tube on the instrument, and then walk away. and then it goes through its process, and you don't have to come back until it's homogenized and ready. And then you can clear the sample. If you have cell clumps or debris or anything, it catches this. Then you spin, and then you have your homogenate. It's a really simple, really easy, really nice process, especially with home brews. The postdocs coming up with the home brews. I was a postdoc. I did this. I had ways and tricks of making the home brews work, and you try and pass that knowledge along, to a student that's wanting to come into the lab, and it doesn't always necessarily happen. So that home brew isn't always reproducible, whereas this is all validated and reproducible. So just some of the features and benefits. So I talked about simultaneous mechanical and enzymatic digestion. It's gentle, and with the GentleMax Octo, it's automated and standardized. It's fast, and it's a closed system. The tubes, I know some labs use the tubes more than once. I'm not going to yell at you, but usually you only use the tubes once with the tissue, and they don't leak. They hit the. Actually, I think I'll get to that, but. And then the benefits, it's gentle, maintain viability with the tissue and the cells. You get good yields, it's reproducible, it's quick, it's easy, it's smooth, and it protects both the user and sample from infection or contamination, respectively. I should say infection. And then some features and benefits with our tissue dissociation kits. Again, pretty much the same thing. They're optimized and pre-titrated, so you don't have to do any kind of R&D work on our kits yourself. Designed to preserve those cell surface epitopes I mentioned. Defined enzymatic activity, and there's lots of consistency. Benefits, you don't have to do any validation work with our dissociation kits because that's all been done for you at Miltenyi. Epitopes available for downstream assays like flow, cell-seq, and some of the assays that Hadley was speaking about. No lot-to-lot validation needed. So I kind of went over our octo dissociator with heaters. Basically, this is our top-end instrument. Processes up to 8 samples in parallel. It's fully automated. It does everything for you. And again, you can run 8 different sample types at once if you wanted to. And you can also have your own user-defined programming on it. So if you want to come up with your own, workflow or your own program, you can do that. We also have the Octo Dissociator without heaters, and I rarely see people buy these because, if you're going to spend, to buy an Octo Dissociator, you're going to spend a little bit more just to have the heating mechanisms, because if there's no heating mechanisms on the instrument, there is a workaround, but it's just a little harder and it's not as automated. And then we have our semi-automated tissue dissociator. So this is what I call the little guy. So this is just a 2-headed simple dissociator. You can only run one program at a time, and it has 2 different heads. So the workaround this for the no heat. So some labs will buy this because there's just not a lot of funds available, but they really like the the dissociating. So we have our max mixer. So you could put the C tubes in the max mixer and put this in an incubator, and it's battery operated. There's no wires coming out to plug in, so you don't have wires coming out of your incubator. So you can get that 37. You can reach that 37 degrees in activating the enzymes without the heaters on the dissociator. But again, there's only 2 samples that you can run in parallel. And there's only one program at a time you can run with the 2 samples. You can't run 2 different programs with the 2 samples, only one program. So there's free programs and software updates for these instruments, and I actually have them. I'm actually going to come around to a couple labs later and update your GentleMax. So if you have a GentleMax and it's several years old and you want me to update it, just come up to me after and let me know, and I'll do that for you for free. These take a USB. It's really easy to do. These take cards. It's kind of a tedious process, but that's okay. So our GentleMax tubes, I kind of got into that. Our C tubes are for cellular functions. Our M tubes are for molecular. So I'll just kind of skip that. It's not very important. Again, our tissue dissociation kits are very easy to use, highly purified enzymes. We also, again, we have our smart strainers, and then we have our dead cell removal kits. So if you have apoptotic cells, dead cells, dying cells, cell debris, these kits will remove that before, so you can just have all live cells, so your viability will be really good. Again, I mentioned our smart strainers that remove cell clumps. And then our tissue dissociation recovery, GentleMax versus manual. You're usually going to have a better number of cells when you run our GentleMax protocol versus your manual methods. Some researchers will have similar results compared to our GentleMax, but then again, you don't know what you're doing to those cells downstream, what assays you can run with those cells, because you don't know what epitopes are left. Again, versus a bead mill, again, the GentleMax outperforms the bead mills as well when it comes to the cells. And then this is our whole list of tissue dissociation kits. So again, I mean, pretty much any type of tissue, any organ, we have a dissociation kit for it. And we also have our newest kit, FFPE tissue dissociation kit, but that's only available to use on our GentleMax Octo with heaters. It's not for use with our dissociators without heaters. And then again, on some of our tumor dissociation kits, we have epitope preservation lists. So, if they're in black, they're stable. If they're in red, they have a strong sensitivity. So the tissue dissociation kit is probably, you might not want to use that tissue dissociation kit with that particular homogenate if you want to, preserve a certain type of epitope. So we have all this information for you guys to look at. And then I'm just briefly going to go over cell separation, because that's one of our biggest things. Our two biggest portfolios are our sample prep, which is our GentleMax, and our cell separation, which is our MAX, which is our magnetic cell separations. So basically, it's our microbeads, columns, and our magnetic separators. It's really, really simple. So we have nano-sized microbeads, and with our column-based products, our microbeads, our nano-sized beads are conjugated to antibodies, and those antibodies will bind to the cells that you are interested in isolating from the homogenate. You could do positive isolation, depletion. We have untouched isolation. We have a bunch of different types of isolations you can do depending on what you're interested in. So this is just a picture of a cell, and this is our microbead conjugated to an antibody. So they're only 50 nanomicrons in diameter, so they're very small. They're super paramagnetic, and they're colloidal with a high surface area to volume. So colloidal meaning that they're always in suspension. You don't have to, vortex them to get the beads in suspension, and they're directly conjugated to a monoclonal antibody. So when you're, and then our column technology, we have our magnetic beads in our columns, and then the microbeads bound to the cells will flow through the column, and all these magnetic beads in here creates an even stronger magnetic field, 10,000 times stronger with the inclusion of our columns. Once you have our microbody or our microbeads bound with antibodies, and you put our microbeads in solution, they're going to bind the cells, and the cells are going to be held in suspension. So this is just a quick look at our microbeads. This is our column. So these are all the little cells with the microbeads and antibodies held in suspension within the column, and these are the beads in the columns. So as you can see, they're held there by a strong magnetic force. And then once you take away the magnet and elute, then all of our cells are eluted. So here's a couple of our, well, here's all of our columns actually. So we have our positive isolation columns, our depletion columns, and then we have our Automax, which is our automated instrument for cell isolation or cell separation. And then we have our molecular columns, mostly for mitochondrial or nucleic acid isolation. But they all obviously come with our paramagnetic beads that create that really strong magnetic force to hold all those cells in suspension. So what's great about MAX technology is that because our microbeads are so small, they only take up around 30% of epitopes on the cell. So let's say you're doing a CD8 isolation. Only 25 to 30% of those CD8 epitopes are going to be bound by our microbead antibody complex. So that means that all those epitopes are still intact for downstream applications, flow, stuff like that. We also have our untouched isolations. It really truly means untouched. So basically we label everything that you don't want, and that comes out. And then you have nothing but the cells that you want that are truly untouched. And also briefly, since our microbeads are so small and we don't need many microbeads because our magnets are so strong that you don't have these aggregates like you see in some of the competitors that have column-less technology. You have these strong aggregates that really changes what the cell looks like and what the cell is doing. And they take up a lot of epitopes, so you don't always necessarily have those epitopes downstream also. With our microbeads and our MAX technology, you have no cell activation. You have touched and untouched. Full downstream compatibility, like again, no effects on cell biology with the positive and the negative selection. Briefly, just these are our different separators. So basically our separators are our magnets. We have a mini and then an octo. So the octo is basically 8 minis. So if you only do a couple cell isolations a week, you probably want to go with the mini. And if you don't have many cells in your sample, you'd go with the mini and the octo. But if you have a lot of samples, you'd want to go with the octo. And then we have our midi and then our quadro. So the midi, our quadro has 4 midis basically, and that's just if you need more, if you have more cells in your sample, you go with the midi. And the midi, the mini, the octo, the midi, the quadro, they all have their own columns as well. And then we have the VarioMax and the SuperMax separators. So the VarioMax can be used with all the columns all the positive columns that our LS, our LD columns that come with these separators. And then our SuperMax can be pretty much used with any column. But for most purposes, these separators are what is needed for the researcher. And then I just want to talk briefly about our automated and semi-automated separators. So here's the manual. You can use a decent amount of sample numbers, and then your cell numbers. You don't really use that many cell numbers, but if you have a lot of samples, up to 15, 20, 24 samples at a time you have to process. We have the Multimax cell instrument. So basically you have to label the cells yourself, but it uses a vacuum to kind of pull out the fluids and everything from the columns, so you don't have to wait around for these things to drip and collect. And then this one is our automated instrument, which I'm actually doing a demo in Dr. Kaplan's lab tomorrow that I think hopefully some of you are attending. So this can use up to 5 samples, but it's completely automated. So all you have to do is put the sample in and put the reagents on and walk away. You just have to input your volume, and there's a barcode scanner. If you scan, all of our reagents come with a Q code on it. and our barcode scanner will scan the code, and then the instrument will automatically populate what program it needs to run, what clean cycle it needs to run based on what we're doing. It's a really nice instrument. It's completely automated. You throw your samples on, input your volume, and walk away. If you are doing a lot of samples, this might not be for you. But if you're doing 5 to 10 samples a day, and you want it automated so you can do completely other experiments in the lab, this is the instrument you would be interested in. The Multimax kind of combines both the Multimax and the Automax Pro. Basically, if you need a lot of samples, it's totally automated for a lot of samples. This is totally automated for just 5 samples. So that's the difference between the two. So just because of time, I'm just going to skip just to talk about some of our downstream products. So basically, we went from solid tissue to dissociation, which I talked about with our GentleMax, and our cell isolation with our Max technologies. And then you can go to cell analysis, cell culture, molecular analysis, and we have flow antibodies, we have cell culture media, we have cytokines, we have stimulations. We actually have a lot more products than you probably think at Miltenyi. So please, if you're interested, please come talk to me after this, or email me, or call me, whatever. But our two main product lines are our sample prep and our cell separation. So with that, I'm going to give you Egon to go over 10x.
Thank you. Can we ask questions at this point?
As long as they're not that long. If they're long questions,
No, I think we'll open to other people to ask questions.
Okay.
Any questions?
Sure.
Okay, you have talked about a lot of cell preparation. Are those designed for single-cell sequencing or for general application?
Both. So you can use some of our stuff for single-cell sequencing if you, basically with our columns, you're going to have a single-cell population. Right? You're going to have a bunch of single cells.
Because I didn't see any data from you to show your system provide excellent quality of single-cell sequencing. That's what we are looking for.
Right. So that's what the office hours is for after this. So just stop by and we can go over that with you.
So just a related question, have you used the FFPE dissociation kit for single-cell sequencing?
I have not. I haven't really talked to many researchers because it just came out not too long ago. And a lot of the academic researchers don't have the Genomics octo heaters. A lot of the researchers have the smaller dissociators. So a lot of academics haven't really used it.
Is that isolate the cells?
Yeah. It takes the tissue, it gets rid of the FFPE, it gets rid of all the storage, waxy garbage, and you have your homogeneity left over. It's a pretty tedious and long protocol, I'm not gonna lie. But to answer your question, I haven't really seen anything related to that.
What's the time frame for the GentleMax on the cell dissociation?
You're talking 5 minutes to 20 minutes. It's pretty quick. Especially if you're spleen or something, it's really easy. It breaks up really easily. Basically our dissociator is a blender. So you're talking like a 25-second protocol, 30-second protocol. And then with the heat steps, depending on what you're isolating, you could run into a little bit longer heat steps with some of our kits. Especially like our FFPE kit, that'd probably be close to an hour just because of so many heat steps. But yeah, it's pretty quick. It's a lot quicker than you think.
Do you also provide support classes for isolating single cells from, for example, paraffin and other fluorines and simple tissues?
Paraffin we haven't really gotten into yet. That's one of the things that our R&D is kind of focusing on right now. Because we want to go to FFPE to paraffin to all these types of embedded tissues that a lot of these facilities have just stored up over the years that they can access. So that's one of the things that we're getting into with our.
How about blood samples?
What kind of samples?
Blood samples.
Sorry.
Instead of the solid samples, there are some other particular samples, for example, blood.
What samples?
Blood,
Yeah. So, are you are you asking for blood on our dissociation kits or our cell isolation kits?
something like that.
Our dissociation kits?
Yeah, dissociation.
Well, you don't really need to dissociate blood from tissue, right? So you would need.
Actually, in the single cell,
Oh, isolated single cell from the blood?
yeah.
Oh, okay. Yeah, we have a bunch of blood products that can isolate the single cell populations from the blood. We have our straight from blood products. So basically, you can use whole blood and just put our microbeads right into the blood, and it can collect those cells for you. We have our Max Express, which uses our magnets to pull apart our blood in 50 ml tubes. Like we have, and we can come from Buffy coats, we can come from whole blood products, we can come from those Buffy cones that are in Europe. So yeah, we have a lot of straight from products from blood.
Can you use the 2-tube dissociator in an incubator to get the heat?
Yeah. So our max mixer. So if you have our tissue dissociator without the heaters on it, we have our max mixer, and I actually have one right in the corner here. You put the tubes in the mixer, and our mixer is battery operated, so you just put that right in the incubator to get that 37 degrees Celsius for the enzyme activation. If you want to, dissociate without the heater. So there's a workaround for that. You have a question?
I have another question regarding the dissociator. What's the efficiency of those either manual or automated systems?
So, I mean, it's very, very efficient. I mean, I would say more efficient than manual. I've seen, cell viability downstream, like 80, 90%, 95%. But it just depends on, if you have good tissue you're putting in, though. I mean, if you have junk you're putting in, you're going to get junk out, right? So it really depends on what your starting material is. If your starting material is viable and the cells are not dying and they're healthy, you can get very good viability and efficiency using our systems. And all that, we have a lot of stuff online, a lot of application notes, a lot of white papers online, talking about our different dissociation kits, things like that. All right, thank you so much, guys.
Thank you. Well, hello everyone. My name is Egon. I'm your local FAS, Field Application Scientist. Actually, kind of local. I don't live here. I'm based in Chicago, but I cover this site. And if you have any questions about sample prep related to 10x or how to run an experiment, feel free to send me an email. I'll be more than happy to partner with you and provide you suggestions, insight on how to properly prepare your samples. So what I want to talk to you today about, it's basically actually something that Yulong and Hadley already mentioned. Sample prep for 10x is critical. It's by far the most critical part of the entire workflow. The workflow itself, when you get to the 10x experiment, is pretty straightforward. It's streamlined and Link's laboratory is being trained, and they know very well how to run the chip. But before getting to that point, you basically need to prepare your cells, and really sample quality is absolutely critical here. If your quality of your cells is low, or if you have a lot of dead cells or a lot of aggregates, clumps, debris, the data quality at the end of the experiment, and that means after running 10x and after spending time and money on the sequencing, that data would not be, unfortunately, would not be very good. So what we really want to focus today is basically, so those are actually the slides that we showed during the training. So the first thing is we really want to start with a very clean, nice, and homogeneous single-cell suspension. We are running single-cell RNA-seq experiments, and so we really want to start from a single-cell suspension. And there are challenges associated with that. So first of all, dissociation, so just as Jim already talked about. We want to avoid presence of clumps, debris, and of course, free-floating mRNA. This is what basically is leaked from dead cells or dying cells. That mRNA will be partitioned into gems. You will be making a library out of that mRNA, and at the end of your assay, the background of the assay will be high because the mRNA will be pretty much scattered all across your partitions. Then we really want to start with a very high viability cell population. As a 10x, we recommend starting with at least at 90% viable cells, but I do understand that might not always be the case, especially when working with tissue. We do have many customers who have been getting good quality data with 80% or plus viable cell. But if you can get up to 90% or above 90%, that is where basically our recommendation falls. And so this is really what we recommend. And of course, again, there are challenges associated with getting high viability. And the third and last point is that we really want to handle our cells very, very gently, which means we always recommend actually using wide bore pipette tips when resuspending your single cell suspension and when washing your single cell suspension. Those tips are available from Rainy, and basically they have a wider orifice that induces way less stress on the cells, and we know that they induces much less damage on the cells. And so this is basically what we start from. And then once you process your cells, it's always important to choose an appropriate buffer. And I have a couple of slides showing also what are the buffers we recommend. And time is also a critical factor. We recommend, once you have your single cell suspension, we recommend keeping the cell suspension on ice for half an hour, no more. And within that half an hour, you should bring your cell to Link, and they will load the cells on the Chromium. So this time actually might vary depending on which cells type you are running. There are some cells that are gonna die or clump within even less than 30 minutes. Some other cells, like tumor cells, they are pretty sturdy and they can survive for hours on ice. So the time is really dependent, but we recommend basically once you have your single cell suspension, keep them on ice and try to get to Link's lab within half an hour. So this is our support protocol, is support.nextgenomics.com. There are a lot of demonstrated protocols and tech notes that are available for download, and they're for free for everyone. And I will actually start with this flow chart. It's a guidelines for optimal sample prep. You can find it on the single cell gene expression tab. We basically have general sample prep guidelines that are they were basically developed in-house, and those are more for like a general sample prep type of samples. Then we have more specific sample prep demonstrated protocol, and by the way, we use in many of our demonstrated protocols we use Miltenyi kit. Like we have a dead cell removal protocol, which is here. Which uses Miltenyi beads. We have a protocol for CD3 positive T cell enrichment, and again, also that uses Miltenyi beads. We are very happy with those products. And then when it comes to like some specific sample types, we have a protocol for dissociation of mouse embryonic neural tissue. We have a methanol fixation protocol that was released a couple of months ago, and then we have a nuclei isolation protocol here that is becoming more and more popular, especially when you're dealing with a sample type that is difficult maybe dissociate, or when you're using very sticky cells, or when cells are extremely large and they might not go through the microfluidic chips. We can talk about that later, or if you have any question, of course. So again, many demonstrated protocol and resources on our website. What I always tell customers is that even though we validated protocols in-house, we cannot, unfortunately, test too many cell types or too many conditions. We do our best, but we can only we have like a limited manpower. But when it comes to your cells or your tissue, you're gonna be the real expert. And so I always tell my customers that they're gonna be the real expert. Our demonstrated protocol provide a very good starting point for sample prep and for general recommendation for when it comes to 10x experiments. And so I will always start from those resources, but then again, you're gonna be the real expert. And so it is always possible that a little bit of optimization might be required. So just to take an example, our nuclear isolation protocol has been validated on PBMCs and mouse embryonic and adult mouse brain. It works on other cell and tissue type, but again, optimization might be required depending on what samples you're gonna run. So again, those are our general recommendation. Is something we always mention during the training, and we really spend a lot of time talking about those recommendations in the lab, and then on web meeting with customers or on site visit. Again, the very first thing is we really want to start with a nice, homogeneous, fully dissociated single-cell suspension. If you see clumps, if you see debris, you will need to take action. You will need to try to remove them, either by straining or by doing a couple of more washes. I'm going to talk about that in a second. The second thing is the viability, as I was mentioning before. 90% viable cells is what we recommend for optimal results. If you cannot get to 90%, let's say if you are above 80, might still be okay. And then if your viability drops, we have our demonstrated protocol for dead cell removal that uses Miltenyi beads. And I was mentioning cell handling is super important. You don't wanna. Here is basically where you don't wanna pipette your cells very harshly. What I will do is, once you have your single-cell suspension, always use wide bore pipette tips and take 3 seconds to aspirate the single-cell suspension into the pipette tip, and then another 3 seconds to release it. It is slow, but it's basically what it preserves cells and avoids cells from being lysed or damaged or completely burst. Our typical recommendation buffer, our typical washer resuspension buffer is PBS with 0.04% non-acetylated BSA. This is not the only buffer you can use. Actually, I can say if you have sticky cells or if you think that the viability of your cells might increase with a little bit more BSA, you can definitely increase the BSA concentration up to 2% without any negative effects. And so typically when a customer tells me that cell tends to aggregate or clump, I tell them to try to increase the BSA concentration to 1% or up to 2%, and you won't see any negative effect on the performance. So yes, if you see debris or aggregate, try to remove them by either washing or straining them using filters. And we have a recommendation when it comes to filters, which, by the way, one of those filters we recommend are from Miltenyi. So we work nicely together. The other very important thing is cell count is critical. So if you target 5,000 cells for recovery, you do need to have your cell stock concentration calculated accurately. In-house, what we use is this the count S2, but I I have many customers using the old-fashioned hemocytometer, and as long as you use it within its range, it's super precise. And so, my message is, no matter exactly no matter what technology you use, but as long as you use it within the range, you should be fine. Now, we recommend providing the cells with a concentration between 700 and 1200 cells per microliter, which is 700,000 and 1.2 million cells per ml. This is you will need to measure the concentration in your lab and then bring the cells to Ling, and they will check the concentration again and the viability again. This is basically a range where we know that the recovery will be optimal. And this is basically what allows you to recover between 500 and 10,000 cells per channel. So always, yes, count cells very accurately in determining viability. And yes, the last point, as I was mentioning, once you have your single-cell suspension ready and you determine your cell stock concentration and viability, keep the cells on ice and try to run them within 30 minutes. So this slide here, just for a second, ignore those plots here. This is something that we described during the training, and it's basically how your data will look like, how clean your data will look like. And just focus on this row here, which basically indicates the background of your assay. So what we did here was basically to take the same sample and treat it as we recommend. So pipette it very gently using white bore pipette tips, keep it on ice, and then the same sample was treated more and more roughly. So by using white bore pipette tips, or by using regular pipette tips, or even then we try to vortex the cells. I don't know who does that, but we try it anyway. But here, so the take-home message is that as you treat the cells more and more roughly, the percentage of reads that are coming from those little partitions with cells inside decreases, which means that more and more reads are coming from either empty partitions or just from partition containing background. And so at this point, you are moving from a single-cell RNA-seq experiment to a bulk RNA-seq experiment, a very expensive bulk RNA-seq experiment. So this is basically emphasize the reason why we really want to treat cells very, very gently. So those are the cell types we have validated in-house so far, and as time passes, we're gonna validate more and more cell types. The reason why I wanted to show this slide is just because I wanted to mention that we have, so far we validated cells up to 30 micron in diameter. I know that cells a little bit larger can still go through, but again, this is what we validated in-house. If you have like cells around 40 micron, they might still go through. If you have cells that are much larger than 30 micron, let's connect with me. One of the things I would probably suggest is to try the nuclei protocol. In that case, the size would no longer be a problem. And we don't have a lower limit, so no matter how small the cell is, how small the cells are, they will still be partitioned. And there is no bias toward cells with different size. So if you have an heterogeneous population, there is no bias toward partitioning smaller cells compared to larger cells, or vice versa. And here, I don't know, I'm not sure if you can see those pictures very well, but it's just to say, well, this is the single cell, the level of single cell suspension we really want to see under the microscope. So no clumps, no debris, no aggregate. Now, we typically recommend washing, once you have your single cell suspension, we typically recommend washing the cells twice. This typically increases the percentage of viable cells, like you can see here. And then if you have a very abundant samples and your viability is pretty good, I would not do more than two washes. Then at that point you start losing more and more cells without gaining anything in terms of viability. Of course, if your viability is very low, let's say 60%, 50% or so, think about the dead cell removal kit from Miltenyi. Now, this is what we recommend when you have a lot of cells, let's say 300,000 cells or so, starting with. If you have a very limited sample, let's say you have only 20,000 cells, or 10,000 cells, or 30,000 cells, you cannot do multiple washes, you're going to lose cells. So what we recommend is to basically check the concentration in the cell suspension media, and then just spin them down, remove most of the supernatant, leave maybe 50 microliters, and then resuspend the cells in that 50 microliters and load pretty much everything on the system. Now, it's not ideal because you might not know exactly how many cells you're loading, and you might not know what the viability is. But if you have a sample which is very limited in quantity and it's a very precious samples and you still want to run it, I always say it's probably better to run it rather than lose this precious sample that might be coming from the clinic or so. So those are the other media, the alternate buffers or media that we recommend, that we have validated and you can use as a resuspension media or washing media. When it comes to cell culture media, you pretty much can use any of those with up to 10% FBS, and we haven't observed any decrease in performance with minimal reduction or no loss in performance. And this is one of my last slides. So if you see debris, those are the two cell filters or cell strainers we recommend using. One is from FlowMe. One is from Bellart, and they are those FlowMe cell strainers that they basically fit the pipette tip. And they're they work really well when you have a small volume to work with, like something like 500 microliter or 1 ml. If you have larger volume, in-house we routinely use a max strainer from Miltenyi, and they also work very well. And again, use them and pretty much debris should go away. Yeah, and just to conclude, we have multiple type of resources on our website, and they're all free for everyone. We have how-to videos and training modules and scientific seminar. For those, you will just need to register, so enter your information, your contact information, but you can opt out from receiving marketing emails. So we don't want to spam you with a lot of emails. I think that was my last slide. Yes, because the next is a blank slide. Do you have any question? Thank you. Yes.
So you can remove your dead cells and debris, but what about the floating RNA?
Good question. Yeah. So typically, again, a couple of washes is enough. Let's assuming that your cell viability is above 80%, close to 90%. Typically, a couple of washes is enough, and you won't expect much background. Actually, background should be very, very low. Now, if your cell viability is low, and you might need to do more washes to both increase the cell viability and remove free floating mRNA. Another situation I can think of is, let's say you use a red blood cell lysis buffer, and you get rid of all red blood cells with a 10-15 minutes incubation. Now, whenever you use a red blood cell lysis buffer or similar buffer, you will need to wash your cell pellet multiple times, because it's true that you got rid of your red blood cells, but the mRNA is still there. And so, yes, this is something that you probably you will need to do. Likewise, if you use DNAs during your tissue dissociation, again, make sure that you wash your cell pellet multiple times, not only twice, but multiple times, to just remove all traces of DNAs. Otherwise, if that is partition, it's gonna chew up the cDNA. Yes.
Multiple times being 3, 4, 5.
Yeah, I would say so. Typically, it's 2. I would say if you can do at least 4, that is, yeah, do go for it. Yeah. Yes.
Is there a type of DSA to recommend purchase?
Yeah, we have so we have a recommendation on our user guide, and so there is a table in the introduction. One BSA, one of this item is from Sigma, and it comes when it's a 10% solution in PBS, and the other one is a powder. But yeah, we have a recommendation on our user guide. Yes.
I have a question about that. So, you recommend to use 10% BSA for the median. So, do you think if you increase that concentration, like 20%, 30%, will affect the result?
It might, yes. We have not validated media with more than 10%. It might affect the performance of the reaction. Yes.
So that's not going to use more than 10%.
You can go up to 10%. You can go up to 10%. Yes.
I have a question regarding the network removal. I think at one time we loaded 10,000 cells, but in the sequence we can only see like 2,000 to 3,000. What could be the problem?
So do you know what I mean, depends, because you use the dead cell removal kit, but then then I guess you loaded the cells according to a specific concentration.
We know we loaded, up to 10,000 cells, but in the result we can only see like around 2,000 to 3,000. Why we lost the cells?
Well, cells are probably dying.
Yeah, that is a possibility.
Either dead, dying, or starting to die.
Or our cell counter may not work well. So there could be multiple reasons.
There are multiple reasons. So yes.
Some people try to use DNAs to treat the cell to avoid aggregation. Is that a good idea?
We haven't tested in-house. We don't use DNAs. But as I was mentioning, if you do use DNAs, make sure that you wash the cells very thoroughly. Otherwise, the DNAs will stick in a partition and it will basically digest your cDNA as it gets synthesized. Yes.
So just to follow up on what we talked about earlier, have you guys done any formalin fixed cells?
We haven't tested it, yeah. Not yet. We did methanol fixation.
You have a lot of formalin-based issues?
I saw that you had a couple of notes regarding FACS sorted cells.
Yeah.
Any additional notes for sorting out a small population that you would like to consider?
So you can load cells straight from the fax if you want to. That is doable, and we have many customer fax sorting cells, and then what they do, we always recommend to recount the cells, because depending on which fax machine you use, you can have like a 20% of overestimation up to like 100% overestimation. So always count the cells after fax sorting them. But yes, depending on how many cells you have, and they might just load them straight from the fax.
But what concentration do they need to be at to load? Because would fax sorting directly into the buffer be too dilute?
It depends how many cells you're gonna fax sort. I mean, that's a possibility. If you fax sort, if you have a very rare cell population, they will be very diluted. At that point, it's always a question of whether you can concentrate them, or if they're all you can get, just load them. And yeah.
A question about viability. You said here is 90% viability, but actually majority of the cells has been sent to us cannot reach to that.
Yeah.
What's your recommendation?
Yeah, I mean, this is what I was saying. So 90% is the ideal number, the best.
What's the lowest?
So personally, so this is not a 10x recommendation, it's Egon's feeling. So
we were recording this.
Yeah. So this is not 10x recommendation. So I typically say if the viability is above 80%, we see good data. If the viability is between 70 and 80 is where I start getting a little bit concerned. I would always, so if your viability falls between 70 and 75, let's say, I would expect some background in your assay. At that point, it's a question of whether you want to go ahead or not. If something that the sample is precious and you want to go ahead, or you prefer preparing it again, it's up to you. It's up to the clients. If it's below 70%, you will get background. So, I mean, there might be instances where, you know, someone brings samples at 50%, and this is a super precious sample, and they say, we know we're gonna have background, but I cannot get anything else, and I do want to see the results anyway. It's their call. They want to get, they will get background. But
and there will be a bill associated to that, even if the data is not very good. So, is this our lunch? Is this our lunch?
No.
Oh, don't eat that. I want to make sure that. So all the scientists will be here throughout the afternoon, and the room is R3C428 on the fourth floor, just outside of the core. But before we close this, I want to talk a little bit about more logistics. So if you want to do the single-cell RNA sequencing analysis or any type of single-cell analysis, you need to contact us way early than your cell is ready. So we cannot say, come here to say tomorrow we'll have some cell ready. We cannot do that. And the reason for that is there will be multiple labs. We have dozens of labs working with us. So we have some scheduling as well. And as you can imagine, this experiment is not something that you come, you bring the cells in, and we can immediately do it. We have to do a lot of preparation before your cell arrives. So we need to schedule at the time, at the resolution or accuracy of at the hours. So we need to know that what time of the day you bring your cells in. So do meet with us a couple of weeks before your experiment is planned. And also it will take another couple of weeks for the I mean, even after we went through the 10x protocol, it will take another couple of weeks to do the library prep, sequencing, and the analysis. So if your grant is due in 3 weeks, don't plan to put the data in the preliminary data. So it's just not possible to happen. And another thing is, if you say you are coming at a certain day, at a certain time, and actually we will have 1 or 2 people specific waiting for it. We have to stop all the other experiment for that person. So it's a big obligation. So far, we're not charging that if you cannot bring that in, but we will record the credit, and so you may be charged in the future, and we'll figure out how to do that. Another thing I want to mention is we're always here to help, and the link is always very much accessible in the lab. If you are not sure whether your cell preparation protocol is good enough, and we recommend you to do a couple of rounds of testing. Okay, without I mean, if your samples are clinical samples, there's nothing we can do about that. We have to do whatever we need. But if it's a cell-based or it's an animal model, so if you can spare some of the samples, and we recommend you to do the testing, and then bring your cells to our lab, just pretending we're going to run. And then we will check the viability, check the quality, check the concentration, check everything, and when it works, and then for the real deal, that we can making sure that increase our success rate. Okay, what else? So do contact us a couple weeks before the experiment, and we need a couple weeks for that. Okay. Lynn, is there anything else you want to talk about? Okay.
If you were,
Yeah.
If you are interested in talking to Jim and Eva, please come over to schedule time.
Yeah. Okay. All right. There are some. That's not our lunch. We just make sure that don't eat that. Okay. Thank you very much. So